Hello, everybody. Welcome to wherever you are. My name is Vinayak Dravid. I run the Nuance Center, which is our facility infrastructure here at Northwestern. And also this event is a very special event. It's called iNano. It's under the auspices of our NSF center called SHINE Resource. And there'll be a quiz at the end to describe all the acronyms. SHINE stands for Soft and Hybrid Nanotechnology Experimental Resource. And we're very excited to bring together Chicago region folks and colleagues. So iNano is special in many ways. It's a coordinated effort among Northwestern, Argonne National Lab, UChicago, and other regional institutions. So as a part of the SHINE resource, which is our NSF center, we organize outreach events, educational events. We, of course, offer infrastructure. And on the research side, we really try to address what are the infrastructure needs in emerging areas of science and technology. And I couldn't have picked a better group of people today and a topic that's also very special. So today we are talking about an event which brings together folks from Northwestern, Mike Jewett, UChicago, Argonne National Lab, Arvin and Austin. And it's going to be emceed by my great colleague from Anthropology, Sarah Young. And the philosophy behind this event is really to bring up ideas together in the region and how they relate to infrastructure and all the innovation paradigm that are taking place. And I was actually looking forward to this specific topic of great personal interest. So here we have three areas that are converging. You have a synthetic biology, as most of you know, is an amazing area of growth this century. I was told that this is the century of biology. I was also told that this is the century of data science. So we have another area that we'll be addressing today is data science and how synthetic biology, data science converge with respect to one perennial issue we always face in humanity is ethics and the societal impact. So I'm very delighted to welcome all of you to this very special event. And I'll pass on the baton to Sarah Young, Anthropology Associate Professor at Northwestern, who will take it from here. Sarah, it's all yours. Thank you.
Thanks, Vinayak. I'm very honored to be a social scientist among a bunch of physical scientists. Maybe I'm a wolf in sheep's clothes. Maybe I'm a sheep in wolf's clothes. We'll see by the end of these couple of hours that we have together. So Vinayak asked me to say a couple of words about anthropology and synthetic biology. And you might say, what the heck? But it makes sense if you give me like 2 minutes to explain. So anthropologists, you may or may not know, it's the study of humans or the study of human cultures. And in this case, this particular anthropologist studies maternal and child health. And it's with a commitment to social equity. So I do a lot of work on water, a lot of work on food. And sort of with this lens in mind, I have brought some cursory understandings, I think, to synthetic biology. So one of our primary methods, anthropologists do what's called participant observation. And so you might have heard of Margaret Mead and what that means. And so she did wonderful studies of understanding what teenageness was like by hanging out with teenagers in Samoa. And so that means you watch what other people are doing and then you kind of do it too. And I have had the opportunity to observe synthetic biology in the wild in my very own house. So Julius Lux is many things, including an active member of the Center for Synthetic Biology, but he also is the dad of my girls. And so I can see how synthetic biology kind of operates and what it's like in the wild. So this is Thanksgiving, where he is testing out, seeing if these kids, including our kids and his nephews, can do synthetic biology tests. And when our kids aren't interrupting us at dinner, we can even maybe cross-pollinate on our research interests. So this is an opinion piece we wrote for Scientific American about water tests. And happily, we're starting to make some inroads. So we just finished a study in Kenya. We were looking at geogenic, so this is like at-home tests to look at naturally occurring geogenic fluoride. We are about to launch on some rapid tests to detect lead here in Chicagoland. And so I can start to see how synthetic biologists work. So it's working, these tests are working and the data are coming in. But who will use these data and for what? For example, in the case of lead, should lead test results be disclosed to future home buyers or other tenants? Can insurance companies use the data to claim that lead tests are a pre-existing condition? How does diet change? So like if you know there's lead in your water, do you sort of shift to soda, sugar sweetened beverage consumption, which has its own issues? And can we prioritize who's getting lead lines replaced using these data? And the time to start answering those questions is not once everyone is using the tests. The time to be asking these questions is now. And that's why I'm so glad that we're here together to be discussing what the implications are for when synthetic biology meets big data. I mean, these are questions to be asked. We're not going to answer all of them today, but I fully expect us to make at least a little bit of progress in that realm.
Sarah, and then if you prefer, I can ask that during the panel. One area, this is the iNano event, Mike and Sarah. So I'm just wondering, can you give us some insight into how Chicago area ecosystem is working out for SynBio, and what are the missing pieces and how we can be competitive as briefly as you can?
Yeah, so that's really great. I'll try to answer just in a short amount of time. Yeah. Certainly something we can come back to in the panel if we'd like a longer discussion. But I think in terms of synthetic biology, the Chicagoland area is really shaping up to be one of the hotbeds. So I think that we have a lot of things in our favor. The Center for Synthetic Biology at Northwestern has grown to be one of the largest concentrations of synthetic biologists in the country and arguably the world. But this is being built up and matched by our partners at the University of Chicago, IIT, and then of course we have great connections to Argonne, and something that has been slowly percolating but perhaps not fully activated is kind of the intersection between kind of Argonne, U Chicago, Northwestern, and what's the special sauce that connects us all in something unique in synthetic biology. In terms of where the opportunities might come from, one is just crystallizing that, making it real. The second is in the context of facilities. We'd love to imagine kind of foundries or tool accelerators that could really facilitate research and projects at a scale that aren't typically achieved in academic settings. And for that, I think you need large scale investment. What's fun is if you did that in a mindful way, it could certainly benefit us as an academic culture, but could similarly benefit startups and help build that ecosystem. The second point I would just say is, we're not the coasts in the context of the biotech ecosystem, but I think all the pieces could be here. We just need to build and we need to keep people here, right? And so I think from my perspective, that's really one of the things to think about. How do you build the like everything from the business and management ecosystem to the depth of kind of researchers that wish to stay here, stay in the Chicagoland area and work for synthetic biology companies. LanzaTech is proving that it's possible as being probably one of the kind of largest synthetic biology centric companies here. And I think they're paving the way and hopefully others can build off of that example.
Excellent, thank you. Back to you, Sarah. You can continue.
Well, I think it's time to hand it over to our next speaker, unless I'm forgetting something.
No, that's perfect, yeah.
Do you want to introduce them, Vinay?
No, it's okay, you can go ahead. Yeah. So thank you, Mike. Maybe I'll just say a few words. Arvind is at U Chicago and Argonne National Lab, and he's a new dad, and we're very delighted to have him in his multiple capacity today. Back to you, Sarah. You can, continue to share the screen. Thank you. I'll see you on the other side, Arvind.
All right, thank you. So I hope you can hear me. Good. So,
yeah, perfect.
Excellent. So what I'm going to talk about is kind of follow up on what Mike was saying, and specifically focus on AI and machine learning techniques for synthetic biology in the 21st century. And something that I'll try to emphasize is this aspect of access. Try to emphasize the aspect of equity as we are trying to build all these techniques. And so I don't have to introduce, Mike already did a fantastic job of, it's very hard to follow Mike in terms of work that he's been doing and pursuing. And what I'm going to say is that everybody uses a phone, but much of the techniques behind the phone and much of the technology behind what's being happening on your phone, by getting recommendations for movies or getting recommendations for restaurants or driving paths through the different parts of the country and so on, everything happens via this thing called as machine learning or AI. And there's a distinction between machine learning and AI in most of the community, but I'm just going to focus on machine learning and basically say, how do we define machine learning systems? So Tom Mitchell, who taught me machine learning at Carnegie Mellon, he defines machine learning as basically a set of computer programs that deals with the construction and study of systems that learn from data. So it's kind of easy to say the statement and leave it at that. And if you start thinking about it, already lost, how do you learn from data? What does data tell you? Is there something hidden in the data that we are not using and so on? So a lot of the information that Mike was able to talk about today is the way they're generating data in biology is just exploded. And what we need are computer programs that can learn from experience with respect to some class of tasks and measure its improvement over the course of actually adding more and more experience to it. So this is typically how we learn in the real world through experiences that, if you touch something hot, you're going to get burned. And, if you do something bad, you're going to get a return on that. And so it's like learning through that experience, but you want to do that in a program. And a very traditional view of how computing was for the longest time is that you had data, that is inputs that you defined into your program. You had the computer do some things and it basically output your stuff. So this is the traditional paradigm of computing, but machine learning and AI generally tends to deal with the opposite side of this. You have the data and a certain output. You're trying to build a program that dictates the logic behind why that experience led you to actually believe that this is the output that you care about. So this paradigm shift of how we think in terms of data and how we learn from data defines the whole world of machine learning and AI. So if we use that and look at how AI has influenced the real world, people have heard this many, many times. It includes the IBM Watson. Very recently, there were computer programs like AlphaGo that were being played. In fact, the computer bet human players in the game Go very nicely. There's deep speech, where people have been using AI to kind of come up with ways to assist us in terms of day-to-day work. In medicine, there are ways in which you can actually use a cell phone today to kind of focus on a part of your skin or something where you're looking for cancer lesions, and it's able to basically find those things out in a nominal fashion. And you have self-driving cars that are making inroads in the Bay Area and other places. And then you have things like, ImageNet and things like that, where you can actually identify things that might be of interest to any person. So the real world is filled with so many applications where it's kind of interesting to see where AI vignettes exist for very interesting and important problems that are out there. It's also true that more recently, it's playing a big role in science. So if you look at AlphaFold, which is actually an AI that predicts how a protein should fold from its primary sequence. And again, I'll talk a little bit about protein structures a little later, but the idea that you are able to predict this entire 3D fold or 3D structure of a protein given a protein sequence kind of revolutionizes how we think of, having access to data sets. Because if you look at the number of proteins that we have access to today, it's in the billions. And if you want to think of how you should look at these structures, it's very hard to do that even through experimental structure determination techniques. It's very, very hard to do that. So AlphaFold offers a vignette of success in terms of how AI represents a new wave of applications that are coming about. More recently, there has been a lot of work in neuroscience where they have shown that artificial intelligence techniques can be really useful in understanding how the brain processes information. There's applications in nature medicine where they showed that you can accelerate diagnostics for a variety of different medical conditions. And so it's been playing a major role in drug discovery, synthetic biology, materials discovery, astrophysics, high energy physics, you name it. Almost all fields of science are now influenced by AI. But there's, at the core of this, there is something called as a scientific process, which is, how we think of learning from experiences. There is a scientific process of actually how we make these new discoveries. And that's actually something that starts with making some observations. You ask some interesting questions about why do we see these observations? And then you formulate certain hypotheses, which tells you, this is the phenomena that I'm interested in. I want to basically formulate something about this pattern that I've observed and can actually reason about it in some way. And then you develop what are called testable predictions. In some sense, it's basically a falsifiable statement that you go after and test and see whether that particular testable prediction holds. And you start gathering relevant data for all of these testable predictions. That comes from literature. You have to get experiments designed. And if you ask Mike or if you ask anybody in the lab that's actually working on biological experiments, they'll probably tell you that this comes over many years of experience. And there's something that's kind of interesting about the scientific process also is that sometimes, accidental discoveries are the ways that we have seen huge bound and leaps in terms of, how science has actually progressed. So this is usually referred to as serendipitous discoveries or serendipitous knowledge that we gain while doing some experiment. You realize a certain failure and then you try to understand what goes on. And then you're like refining that further and further and generally understand a new phenomenon that you might have actually not started with in the first place. And that goes back to actually feeding this entire loop, and that's how the scientific process actually really works. Unfortunately, when it comes to biology, there is a serious aspect of trying to put together various different things that have to come together. And I like this cartoon illustration that kind of illustrates to us as to what's going on, which is a matter of fact, way in which how biological experiments or chemistry experiments are put together, in which you have various steps that are actually like put in tandem. It's very hard to kind of even try and understand whether we can reproduce the research that's being done by various different groups. There's something that you have to be constantly aware of, which is many of the experiments that were done in the past years have suffered through this aspect of reproducibility, which makes it very hard to kind of say whether our research processes themselves are appropriate or are we following the right set of tools and techniques, standards. So when it comes to this, when it comes to this computer science as a field or as a domain has really been after this reproducibility aspect quite from the beginning. And that's actually starting with this fact that you had hardware chips that we were manufacturing. And many years ago, if you manufactured a chip, there's no way to actually say that this would work in the way that you expected it to work. And that garnered this idea of verification and validation from the beginning of the domain of computer science evolution itself. So when the artificial chips or computer chips were started, manufactured, there was this whole procedure of understanding whether these hardware technologies would work the way that they were designed to do. So it's usually verification, and validation comes across in the place of actually like trying to say, okay, once you design it and once it is actually working, you basically make sure that it's supposed to do what it is supposed to do and doesn't do anything else. So this aspect of going from verification and validation was something that was inbuilt into our computer science curriculum, or even the process of actually learning how to do computer science from the very beginning. So computers have this ability to repeat a number of different tasks in different ways, and that you can actually like program them to do certain things. And that comes from the fact that you have ability to like write software source codes and write workflows that kind of say how these things have to come together. So you have object-oriented programming techniques, you have ways to compile the code in certain ways, you have like using the same set of statistics that you use to evaluate the metrics that you need, and so on and so forth. That basically ensures that you have a way to abstract the concepts that you're trying to investigate and eventually lead you to build better programs. And that allows us to reproduce what we have done for a very long time. That's actually the reason why we have been able to stand up such very large scale systems for doing computer research. So for example, in Argonne, we are trying to build the next supercomputer. And despite the fact that we have run into issues in terms of procurements and things like that, the aspect remains still the same, that at the end of the day, when we build Aurora, which is going to be the next supercomputer in the lab, this is going to be one of the fastest machines on the planet, being able to do like exaflop computations, or 10 power 18 computations per second. So reaching that goal would not have been possible if we did not have these types of inbuilt checks and balances to get our code to run. So in biology, unfortunately, this is actually a very hard problem because you're working with so many different conditions. You're working with biological systems that have their own mentality of how they work. And so you have to work with these nuanced aspects of what biology entails. And that gives rise to like a lot of uncertainty in the sort of behaviors that you expect to see. But one of the hypotheses that we sort of can think of is maybe automation in terms of how experiments are designed and run can come to our aid. So this is something that in the cell-free systems world, or in terms of some of the applications that we are interested in, software tools can actually come in the help of actually putting together different biological circuits and interpret them in terms of experimental protocols that need to be designed. And there is an automated set of construction of these DNA parts, including modules and strains that need to come together, eventually letting them, infect whatever group of organisms that you're planning to work with. Then you can use high throughput screening methods to kind of adapt them in order to understand these, behaviors that you're interested in going after, and use machine learning techniques or AI techniques to come to the rescue in terms of understanding these very large data sets that are coming from high throughput data. Of course, you have to be aware of noise, you have to be aware of biological systems, and you have to be aware of a lot of things when you're actually working with this. But automation as such can sort of be an aid in terms of writing biological programs, eventually leading us to build a compiler for biological programs that might help us build very large scale systems like what Mike was referring to. And the sort of cycle of, trying to do experiments for a very long time, can be very tedious, even on a larger group of people that might be involved. And as a consequence, I think automation, robotics, and AI can come to some aid when it comes to like designing biological experiments that are quite useful. So proteins as such, I'm going to talk about this from the lens of using protein engineering. That's something that we have been interested in from the very beginning. And so I'm going to talk about it from that lens. And although they only have like 20 amino acids alphabets, the combinatorial explosion of how many proteins exist in nature is kind of mind-boggling, right? Even if you take a protein with 100 amino acids, so there's a polymeric chains of amino acids that are put together. Even with 100 amino acids, you can see that the combinatorics is kind of mind-boggling. You have 100 power 20 combinations that you need to explore in order to understand what might that function of that protein might look like. And they perform myriad functions inside of a cell. They're controlled by the location, environment, and other factors. They're influenced by these various different aspects. They take action based on some context that actually depends on what they're doing at that given time. So here is an example where you can actually place different proteins in sort of a nano matrix and kind of convert any substrate to any product of interest by hooking up these chains of proteins in a certain sense. In fact, this was one of the applications that Mike showed you exactly previously, where he showed, how we can produce acetate in this sort of a condition. So they adopt diverse conformations, and they're basically, you can consider them as nanorobots that can be of basic use to any different application that you might be thinking of. And there have been many successes of recent, protein engineering. So de novo design of picomolar mini protein inhibitors, which is something that Mike also referred to. More recently, they have been able to build mechanically coupled axial rotor protein assemblies, which is kind of cool because now you have like mechanically working parts like what happens in a car, and you can really think of these as being applicable to a variety of applications in locomotion or other sorts of applications where you want very little energy being spent for a whole lot of function for the fun of what they're doing. But in all these cases, one of the things that must be understood is the fact that they used machine learning in very small ways, which is that they used machine learning only in terms of analysis of the data, but never in the context of actually like building models that would tell them what is the design step that we need to take in order to get to a particular observation. So that's going to be some of the emphasis of my talk. And I'm going to do that from the perspective of doing antimicrobial resistance. So this is one application where, antimicrobial resistance is a natural process by which microbes evolve to resist action from drugs, making them essentially ineffective. So if you look at penicillin, or if you look at other drugs that are in the market, within about 5 years of their introduction, bacterial systems actually develop resistance. And this is a problem because it essentially renders them useless against microbes. And you have lots of examples of this. For example, multidrug resistant tuberculosis infects more than 2 million people in India, something that I'm worried about, especially if you have to go to a hospital in India, you have to be careful about contracting TB or tuberculosis. And this is a serious problem because it can cause over 500,000 deaths globally every year. And there's very little that we can do in order to prevent them from coming along. And if you pay attention to the number of antibiotics that have been approved in the last 30 years or so, it's actually dropped to about 2 from 2008 to 2012. In the last few years, we have not had any antibiotics that have been approved. And that's mostly because most antimicrobials develop resistance very quickly in the context of bacterial systems. And that's actually something that we all need to be worried about. One of the things that we have been lucky at the lab is that we manage what is called as the Pathosystems Research Integration Center, or PATRIC system, which has over 273,000 assembled genomes, fully assembled genomes. And about 43,000 of them have antimicrobial metadata available, which is kind of a useful way to think of, how can we come up with new antimicrobial peptides that can sort of attack these, bacterial systems. So AMPs are an integral part of the immune system. They vary by sequence lengths. They adopt a variety of structures, including alpha helical proteins or beta strands. They can be mixed or even disordered in some sense. But these cationic AMPs can be potentially powerful as antibiotics because they usually attack the membrane of the bacterial system of interest, and they're going to get rid of the entire bacteria by actually rupturing them. So whereas cell-free systems that Mike was using was for more productive purposes, in this case, it's a destructive purposes, they're being used to basically kill bacteria. And the rational engineering of them requires better understanding of their structure-function relationships, which means that we need to have plenty more data in order to analyze what's going on. So one of the things that we have been playing with in the last few years is to have a lot of models that have come out in the industry. So for example, Google has these models for natural language processing that's basically very good at generating new text. So, if you're writing an email or if you're writing something and you're able to complete the text that actually is based on a model that's called as a large language model, most likely something like GPT-2 or GPT-3. I'm not going to explain all these things in terms of what they mean, but what I'm going to say is that these natural language models have really gone to a point where, question answering or getting logical answers from various, user queries might actually be exceeding what is even possible in some cases by even human standards. So working with these types of models really provides an opportunity for us to see if they can be applied on genetic data. So I'm going to start by looking at malate dehydrogenase family, which is kind of an interesting family to start with, but we started with this family and we said, okay, what does it take us to train these large scale models where we can tokenize them by codons and learn on the genetic data directly. So in some sense, think of it as this way, right? So you can have this natural language processing algorithm that works really well on text. It's able to complete text context and sentences and so on. We said, can we actually learn how to complete a gene structure? So that's the basic idea behind it. And this is something that we have scaled up to like much larger models so far. And the synthetic sequences that we are able to generate now can be folded using AlphaFold to kind of see if they adopt the same structure and they have similar processes and so on. And of course, we need to express them in the lab and trying to verify whether they do the exact enzymatic reaction as they were designed to do. That's actually in process. But when you actually look at these GPT models, yes, we are able to train them on the genetic sequences, which is really awesome. And we can go all the way from like simpler models that just have like 125 million parameters to all the way like 6 billion parameters. And what that exactly means is basically trying to say, how do you represent certain features from the sequences that you can learn from in the data? And when we end up doing that, we can actually see that the learning goes on, the training versus validation steps sort of have their own behaviors. But the important thing is that we can actually now start generating new sequences of malate dehydrogenase that looks like this. So we generated 128 sequences. Don't ask me why 128. 128 is a good number for a computer, but we generated 128 sequences and we started analyzing the sort of GC content or molecular weight and sequence lengths and so on. We were like looking at them. And the cool thing was that it actually spreads out into these 8 clusters without even us saying anything about the sequences. So they fall into these 8 clusters, and these 8 clusters have like very different properties when it comes to like GC content or sequence length. So sequence lengths vary between 310 to about 340, which is usually the size of the protein. You have molecular weights that also vary by this one. And you have isoelectric points also that you can determine from some preliminary data. So the interesting thing was when we were looking for the sequences, we were thinking that it was memorizing the data. And so therefore, basically, it was spitting out whatever it had seen. But no, and it does this thing where there are no duplicates in the training or validation or test sequences at all, which is kind of surprising to us because it's basically learning the underlying structure of what it means to generate a new malate dehydrogenase and builds those in terms of those clusters. And so if you put them back into this AlphaFold program to kind of predict what the structure looks like, now there are 8 sequences, so there are 8 structures that are overlaid on top of each other. But what I want to pay attention to is the fact that there are these blue regions in this sequence, and they've been painted all the way from blue to gray to red. Red regions are the ones that undergo the most sequence changes. So those are all primarily around the region where they're not really interacting with the binding site, which is at the right center here. And you can also see that AlphaFold seems to add other folds into the structure, which we don't know why it did that, but we are able to see that there are new sequences that have like different secondary structural details that are added to it. And these are not happening inside of the protein structure per se. They're happening all the way outside on the surface of the protein. And that kind of gives us some more confidence that it's learning these structures and then adding things based on what it thinks are more interesting aspects that could still be functional. So we are now in the process of like taking these sequences and doing further analysis where we can basically give these sequences that you build and do in vitro validation of whether these enzymes are working the way they're working. But you can also do something where you can build what are called as phylogenetic trees. So that's basically mapping out the evolutionary structure of these sequences that we generated. And one of the interesting things that we observed was that any sequence that you see here in black are the generated sequences. And that is basically synthetic J sequences. And there are a lot of these sequences that are shown here in yellow, pink, and purple, and so on. Those are naturally occurring sequences. And what we find is that basically some of these generated sequences are occupying niche areas within the phylogeny where we have not seen many examples in the in the naturally occurring world of sequences. So these wild type sequences are interspersed between the real world sequences and the naturally occurring sequences and unnatural, which are the generated sequences. And this kind of gives us more confidence that the generated sequences are unique They have their own properties and we'll have to validate them in the lab, but it's kind of cool to see from the point of view of large language models as to how they work. But now let's talk a little bit about applying this to screening of antimicrobial peptides. You typically start out doing this by using what is called as a design, build, test, and learn cycle. And this DBLT cycle is something that you have to design from the beginning. So you start with a place where you can manufacture these peptides, or you can basically generate these peptides. And you do some post-processing and validation to make sure that you get the peptides in the quality that you require. And the AI and ML methods that I just spoke to you about are these generative models that I was talking about, protein language models, essentially. And then you build some of these peptides in the lab, basically by using molecular dynamic simulations that kind of tell us whether these peptides have some actions or not. Basically, you want to see if the peptide will bind to the membrane and it doesn't let go of the membrane. And once you do that, you can sort of characterize certain properties which stabilize the membrane and its interactions. Then you have to do this testing in the lab. But unfortunately, the testing, if I have to get like hundreds of peptides a day, let's say the synthesizer that we have access to is able to like generate hundreds of peptides a given day. If you have to test them in the lab, one person cannot probably do that. And that's where robotics actually helps us, because we have liquid handling systems that can do these experiments. And that's part of reconstructing our equipment of how we think of running these experiments constantly, 24/7, so that we can actually run them quite a bit. And then you learn from this data that's been partially generated, so you can sort of see what's happening. So there's something that Chris Fry in the lab has been pioneering in some sense. So we have this ability to do high throughput peptide synthesis in the lab, which is basically a chem-speed ice swing peptide synthesizer that's present. And after we get this sort of peptides that we say we want to generate them, basically it's a Python script where we basically give him like 21 antimicrobials that we tested last time, but basically give him any number of peptides and he's able to make them as 96 peptides in parallel as basically a 96 well plate. And so there are some aspects of actually worrying about how to do this. There are aspects of cataloging the data, making sure that all the data is stored correctly, and fully automating the entire peptide synthesis aspect is still a very hard thing to do in the lab, because there are some aspects that we need to worry about, making sure that we can purify the proteins, you can do the mass spectrometry, as well as characterization of these peptides as to what they have in terms of secondary structure and so on. Unfortunately, peptides actually have very diverse characteristics. Some of them are soluble, so you can actually see that they're very clear. They have like good properties. Some of them act as pellets, which means that they're aggregating, which is a very bad thing, because it means that we cannot really run our experiments with sort of concentrations. In some cases, they actually form hair gel, which means that they're undergoing self-assembly, and that kind of has a different sort of connotation in terms of how we can run gels and experiments in the lab. So I'm not going to talk a lot about these experiments, but I mean, the generative models, I already gave you an overview of how we were doing that. But the idea that simulations can actually help is kind of interesting. So here is this orin, which is a naturally occurring protein, naturally occurring antimicrobial peptide, where you can see how it actually binds to the membrane in a little bit. So you see moving there, but we have actually studied various mutations on this particular 13-mer protein, where we can see a lot of them actually end up stabilizing the interactions with the binding of the membrane. There are some that don't. For example, this M-lysine 1 does not, and there are some that actually do that. And so it's kind of interesting from the point of view of design, trying to use these MD simulations where we are able to like draw insights on what stability interactions might be, and which interactions, if we modify, we can actually get away with better solutions. And now this generates a lot of data, like 150 gigs of MD simulation data. You need somehow to process all of this information. That's where we wrote the software toolkit that processes all the MD data. In fact, Austin, who has actually done a lot of this work, writing machine learning code for processing MD simulation data and other types of data, lets us actually build these intuitive latent landscapes where we can see which of these interactions are more stable, which of these interactions are not stable. We use that information to guide the next round of experiments, really speaking. And so you have this entire platform that we needed to set up so that we could run these protocols inside the lab using Python scripts. So instead of having a human technician or somebody else go into the lab, pipette each of these things, we have protocols that we write in Python programs that are now sent into the scheduler, which manages a network of these robots that are connected together. So here is this Hudson platform where we use our experimental things to be done. There's a bunch of these OpenTrons, which are about $5,000 a pop, but you can run a whole lot of liquid handling experiments there. But what is kind of cool is the fact that we can control all of them by using a single programming interface that offers a unified way in which you encode your protocol, you write your protocol in a particular way using Python, you stitch together a workflow of how that experiment should look like, and then you run that and see whether you're able to get what you were supposed to get from the experiment. So in some sense, it removes the human or abstracts the human in the path of doing these experiments, but it can continue to run 24/7 without human intervention. But at the same time, it's collecting all of the data into these large data sets where we can analyze for like issues that might have happened. Maybe something actually fell off, or maybe we didn't have a proper action and so on. I'll show you some examples of that. And then the machine learning can actually ingest all of this data at the back end and sort of make inferences for saying, how should we select the next set of protocols to run? So the current Python-based API operates across these different instruments that we have put together or chained them up in some sense. And you can see a video of how this is being run. So the protocol itself is fairly simple, not as complicated as what Mike was referring to in some of his cases, but you have the place where you can set up the cells, you set up the treatments, which are basically the peptides that I talked about. You transfer the treatments to the cells and basically, do a bunch of these experiments. And this is last year's experiment where you can see, the robot basically picks up the plate, comes and places it in one place where it has to do this. And I'll just stop at a point where it has to go back and forth in terms of running the experiment. So it's kind of cool to see what happens when it has to like remove the plate top or the lid from the plate. It has to do it in separate options. So this is typically slower than how a human would operate on these machines. But it also gives you a sense of the complexity involved in terms of writing programs for this, which means that you have to be really careful of articulating what are the steps that are involved, including these minutiae where you have to like remove the lid in a certain way so that you don't bang the thing on the floor or you don't bang the samples and they get mixed and things like that. Now you're going to see the micropipette pick up and doing its stuff now. So it's going to come. And all these protocols have to be calibrated every time you're running. So it's kind of, pain in the backside when you have to run these experiments many, many times. But the fact of the matter is that we are able to do these experiments on the fly, and how we design them is actually like completely automated. So yeah, so this is going to be boring when it runs, but the idea that we can run these experiments continuously is kind of cool. But when it comes to like calibration, the importance of calibration becomes quite important. So when we did this experiment for the first time, we obviously failed in some sense. So if you look at the red line and the blue line, these are experiments that were done on 2 different occasions. And you can see that there is hardly any correlation. You expect the behaviors to be somewhat similar, but they are not. In some places, they look similar, but in many places, they are not at all similar. And that's kind of a reason, because what happens is, there are issues that need to be debugged whenever you write a program. So the idea that you're able to automate it is sweet, but the fact that you have to debug these programs to write in a certain way is actually kind of hard. So here comes this issue of reproducibility in science, that you have to be very careful of how you're running these experiments carefully. And so what we discovered at the end of the day was that there was a possible contamination in the samples that we were working with. So we had to do additional checks and basically get our insights organized. And the next time we did this experiment on 823 after doing calibration, you can actually see the results are more or less believable or trustable. So you can actually see that the behaviors are more or less the same way that we expect them to be. So I'm not going to talk about the liquid-liquid phase separation. This is another project where we are using machine learning tools to kind of guide the design of what we call nano compartments inside of a cell. And that's actually using what is called as liquid-liquid phase separation that occurs within a class of proteins called disordered proteins. And these disordered proteins have certain patterning requirements, which gives rise to this combinatorics problem that we were talking about. And we are screening them using automated platforms like these that I just presented to you. The infrastructure is pretty much the same, but we are looking for microscopic details of how that assembly happens. And so that kind of comes together, and we can actually check them in a temperature dependent manner. So basically you're using machine learning techniques to come up with suggestions for the next set of designs that you're looking at. And those designs are being evaluated automatically by using, lab equipment that I just spoke to you about and how they're put together. But important aspect is that we can actually do these experiments in a completely robotic fashion. So this is actually advanced photon source where we have placed instrument inside of the beamline, where the beamline is a place where you actually shoot X-rays out of. I mean, you have other beamlines also, you can shoot other type of things, but you have these beamlines where you can do this. And that allows us to run experiments by simulating them first in this robot toolkit. And once you do those simulations, again, this is going to be a boring video unless you kind of see what's happening. So we can simulate the exact pipette design and design these custom equipment using robotics. And once you get those things to work properly, you're now able to place the sample accurately in position. And after this, we don't write any code at all. What happens is we basically run it in the experiment, and we look at how this pendant drop experiment worked in the this one. So the capillary way of doing it, the blue lines are basically the handmade experiments, and the pendant drop is the automated way of doing it. You can basically see hardly any difference between the two. And that's actually a good thing, because you're now getting better results in terms of the resolution of how you're running these experiments, and we can run these in an automated fashion. There's an actual beamline in the lab where we are testing it. So it's kind of cool to do this in real time. And you can also do this with AR and VR glasses, which is kind of cool for people that have want to experiment with these types of equipment right from their home. And there are a few next steps which focus on data integration, sort of work with, how do we take all of these data sets together and work with them? And all of these things are coming together for what we call as secure biosystems design, which is an integrated laboratory and computational platform that enables the rapid design and engineering of secured biosystems for a range of applications. Of course, there's a collaboration with Mike, which is going on for a little while. We are now writing the renewal part for that. And the whole idea is that we want to create this in such a way that you have ways to manufacture biological parts on demand, including transcription factor variants or bioenzymatic pathways, or even genetic parts that we would like to see. And we would like to use the cell-free systems as a way to test them with high throughput, and emphasizing on the reproducibility part as well as the platform part as to the robotics, how they help us do these experiments on the fly 24/7. And the combinatorics can be explored in a very efficient manner. So in some sense, this is a complete unified platform that we are developing for experimental computational workflows. It uses AI from the beginning. So AI is a first-class citizen involved in the design, involved in the experimentation, involved in analyzing the data and saying how we can run them. And then finally, unfortunately, robotics is still a very daisy chaining effort. If you want to get into this aspect of pulling together robotics and putting them all together, it's still a very, very hard thing to do, because vendors don't talk to each other. There are hardly any standards. And so we are trying to create this as an open source toolkit that everybody can play with. And we now have applications that are now standing up on various different protein engineering tasks that I spoke about, but there is also the DNA side there. My colleagues are involved in it. With that, I'd like to acknowledge a whole bunch of people that are involved in this in various different levels. You're going to hear a lot of the ethical aspects that Austin likes to chat about whenever we are doing synthetic biology next. But computing time was made possible by Argonne Leadership Computing Facility, as well as the IT folks that basically blessed us by saying that we could do these experiments without getting into tug of wars with how the robots would operate remotely, because all of this was done during the pandemic. So thank you very much.
Excellent.
Thank you, Arvind. Golf clap for Arvind.
Absolutely.
It's nice to intersperse each of these talks with a couple of questions, and Vinayak has one that I think is quite interesting. Vinayak, would you like to pose it?
Yeah, very briefly, typical biology evolves over millennia. There are mutations, they get adapted over a period of time. Synthetic biology in some ways gives you an answer, or potentially can give you an answer much quicker in terms of the RNA you can construct, and the ribosome will make the protein of your choice. So how do you reconcile this dichotomy in the context of what you do in a data science? And is there an opportunity to look at intermediate steps or just go to the final minima? So can you shed some light as briefly as you can?
What a great question. Yes, I think there are some aspects of it which are kind of interesting. So when we were trying to learn these rules of what's happening, so I'm dealing with mostly prokaryotic systems or bacterial systems, which is kind of easier to deal with because they have a much simplified way in which they actually handle, gene translations into protein and so on. So one of the things that we have been trying to do with this, I didn't talk about this, but the generative models that we have been building, we are trying to see if they're learning the rules of how they extract the biological principles behind how they have evolved. So when I showed you the phylogenetic tree, which was just a nuanced basis of actually observing what might have happened over the course of many millennia as to how they were evolved, but it still doesn't give us the rules that why certain base pairs are occurring in certain combinations and so on. That's actually something that's tuned by the GC content of the bacterial system that you might be looking at, and various other factors, including environmental and other things that play a role there. So when it comes to those sort of things, what we are trying to do with these language models that we have adapted for these type of toolkits is to try and see if we can extract those biological rules from analyzing these data sets directly. So are we able to extract the rules that basically tell us why a particular base pair occurred in that particular position and so on? That's a very hard problem in general, but the hope is that that's how we'll be able to learn and make the models even better. And that requires these AI algorithms in a way that you can have humans interact with the AI directly, which is not there right now, right? There's a lot of open questions there as to how you make that happen. And at the same time, this also raises many questions in terms of how we are doing this, which is, the data science basically lets you look at the data in various ways and pulls together an approximation of what might be going on in nature. Whether nature does exactly the same way or not is still an open question, which we'll only know after doing sufficient number of experiments, I feel. But it opens as a venue which has not been there for a while. So it's a lot of fun to do this experiments and try and see whether we learn anything useful out of these.
Thank you. Yeah. We'll keep some for panel. Back to you, Sarah.
If there aren't pressing questions from the crowd, I think it might be time to turn the mic over to Austin Clyde, the youngest, but probably the smartest of all of the panelists. And we are excited for Austin to be stepping out into the world to the next phase of his career. And we're looking forward to reviewing all the job offers that he'll be receiving by the end of this presentation.
And we get only 10%, right?
Now, I was going to say that. Okay. We'll share the 10% tonight. Okay. I'll stop talking. Austin, take it away.
Well, thank you for such a kind introduction. Let me start my slides. So,
I'm sorry, but before you start, let me just say, did Arvind want to say anything about Austin besides what he has already said?
So, if at all possible, Rick and myself would like to keep Austin in the Chicago area. And he's going to be in the job market very, very soon. So for a person that actually is like at the intersection of, various different fields, from protein biology to drug discovery to ethics and social concerns and equity in how AI is being applied to biological applications, you cannot find a better expert than Austin. So he is going to be on the job market. He's already a Harvard Kennedy School fellow, and please hire him.
We'll keep that in mind. Thank you.
All right. Well, thank you. So thank you for inviting me to give this talk, and for the wonderful talks from Dr. Ramanathan and Professor Jewett. And thank you, of course, for the Shine Center and Nuance and hosting this wonderful event. It's usually not often that a panel consists of welcoming a conversation about ethics alongside such groundbreaking science at the same time. So, I hope to change your minds on that norm, on whether or not we invite ethics into the discussion more often. And I hope to show you that just being analytic and scientific is all part of the process of communication, of democratization, of fairness, and of scientific discovery at large. And so I posed a question for the audience, I guess, in my title, is how does artificial intelligence change the ethics of synthetic biology? And I'm going to tell you my answer to the question right up front. And I hope throughout my talk, you think about it and see where you might land on it. So my answer to this question is quite simple. I'm going to try to argue that AI can revitalize the ways that we think about risks, the ways that we think about how knowledge might be changing and the ways in which it's flowing. And also to think about how global inclusivity might actually impact ethics. So I was told that a good portion of the audience here might be undergraduates or people thinking about what to study or might just be scientists. So I thought I would just give a little background on how one comes to put together a talk with 3 words of AI, synbio, and ethics all together. I started off as an undergrad studying mathematics and philosophy, which eventually turned into doing cancer research and deep learning because I needed money in college. And eventually I liked the people I worked with a lot, and I thought the problems in deep learning were quite interesting. So I started doing my PhD in computer science, particularly high throughput drug discovery. But recently I've been thinking about how my work and the work of my colleagues might impact the world and society. So I thought, well, I guess I can brush off a little bit of the old Habermas I used to read and go work with Professor Jasanoff at the Kennedy School, working on science and technology studies. These are some wonderful books that I thought were foundational in a way. So, throughout this work, keep in mind that I do still do scientific work, but today I'm talking about my work mainly in the intersection of human rights, democracy, and science. And so just for a little bit of background on this, I did want to throw a slide in of kind of thinking about, we see slides like this quite often that are, large platforms of let's do scientific discovery, and the underlying assumptions of kind of why are we doing it, what's it for, who's thinking about it goes untold. So a lot of my research was around designing computational drug discovery platforms during the COVID pandemic. We were able to find a couple molecules for the protease in an automated fashion. And, one would never question my work and ask, well, I mean, maybe someone might, one would never question it and ask, is this for social good? Are you doing something in making a system that's safe, that's suitable, that won't do any harm? And of course you'd say, no, I'm doing COVID research. Like how bad can that be? But I hope in this talk, you might want to return to a slide like this and start to question a little bit of, well, maybe there might be other things to think about that lie underneath. So today I'm going to start off by giving you a little tour of the rich field of the ethics of synthetic biology. The ethics of synthetic biology has been a field between everywhere in the social sciences to philosophy since the 70s, 80s, 90s. And it has a long history ranging from GMOs to pesticide usages to various fundamental dilemmas in the philosophy of technology. And that's going to lead me to talking about what's called the dual use problem, which is a very, very famous dilemma that technology can't be decided beforehand before we make it, whether or not it's just going to be for good or for bad. And then I want to talk about AI and how AI might push the scales on this problem, and then try to paint you a new picture of ethics that doesn't come from ethicists per se, but comes from engagement of all the different communities that are around for it. So, let me give you the classic example in bioethics, GMOs. If you take a bioethics course, I guarantee there's going to be 2 weeks on GMOs at least. And, one question that you might first want to ask is, why are we talking about GMOs? Well, the pretense for GMOs generally goes with the following, which if you find some random articles on people who are proponents of it, will basically go, there's a food shortage problem that's, arguably already happening and will happen in the future, supposedly. And therefore, how do we fix it? Well, we just need crops that produce more food. And so this, of course, I don't need to rehash, I think, the GMO debate, even though it's been out of the popular media, I think, for a couple years, but there's still millions of dollars of lobbying going on. And even most of the foods that you might buy at like Whole Foods, for example, have like, non-GMO stickers on them, which is always an interesting political thing to think about. And on the flip side, you also have people who argue that, well, GMOs are actually a good thing because we're avoiding the usage of pesticides. And pesticides cause numerous cancers, environmental disasters. I know Sarah here can tell you all about the water problem they cause. And in general, they might be better. And so, in ethics, you have this kind of problem usually. An ethical problem generally looks like a dilemma, which basically says, there's a left side and a right side on this problem. We have this tool, GMOs, which we know can increase our crop yields, can make our food more stable, more secure. But we also don't want things to get out of hand, like the ad of a pig fish going around. We don't want something that might be unnatural or not naturally occurring. Though that's always an interesting question, is whether or not something we make is unnatural. There's, I think, some really substantial concerns about gene editing gone wrong in the sense of it being transmissible, in the sense of terrorism, getting into the wrong hands. And there's also the sense to which, like, we just don't know. It's a very new technology. In fact, the whole theory of how the gene system works is rather new. So, there's reasonable concerns. And on the other side, there's very reasonable, if not fundamental, problems that are tenets of economic and social human rights, such as looming food crisis, pesticides and workers' rights, the basic right to water. And so in ethics, you're presented with then a series of questions, which are, how do you weigh these risks against the problems they might solve? How do you know if you know enough to actually work on solving them? And today I think I'm addressing a room of mostly scientists. And so one thing that you should keep in mind is that, this weighing process of thinking about how do we balance risks and problems might not be your problem necessarily. And so one thing you should think about, instead of thinking about, is this right, is this wrong, how can we weigh this? What we can think about is how can we produce information for others being the citizens that we all live with globally in your country, how they can think about these problems. And I had to add in a little slide earlier of from a comment of Mike Jewett, which I thought hit the nail on the head in some senses. He said, something along the lines of, is it really society addressing, or is it something along the lines of, we have to invent solutions so society can address the problem? And one interesting comment is just to think about, is it really society addressing the problem if we just invent solutions? There's always this question of what's the problem that we're solving and why, and what are the alternative solutions? So on this note of just thinking about technology and the various problems it might present, if we dive into the whole GMO debate, I know there's many different technologies around how we might genetically modify food crops or different biological organisms. Let's focus on one, say like CRISPR-Cas9, which is a gene editing technique. There was a bunch of fear and a bunch of moratoriums placed around research with CRISPR-Cas9 due to the just unknown unknowns about it. And this kind of came into a reality. And just a few weeks ago, the Chinese scientist who used CRISPR on two twins, or a pair of twins, was released from prison. And it makes you wonder that, why are we inventing technologies that lead people to go to prison who claim they're just doing basic science research? And, in Craig Venter's own words, he says, the question is not when, but if are we going to start modifying human genomes. And so the overall problem that this is called in the literature is the dual use dilemma, which is that technology can be used for multiple different things. It can be used for just the things it's intended for, but it also can be used for other things. A very simple example that might be most maybe in our heads is even the car. We take for granted that cars are there to move us, but it's not clear that that's the only thing cars do. Someone from someone may say from the Back to the Land movement in the 1970s and 80s on the West Coast might look at a car and say, what is the function of a car? A car is here to pollute and destroy the environment. Another person might say, who was in France at a very unfortunate and devastating time to the country, would say cars are used as battering rams and political moves. Some people in the United States might say trucks were used in the same way during the trucker protests in Canada. And so just because a car we think is to be used to move, the car has no self-reflexivity to claim what it is for and what it's not for. And so just to dive in a little bit about this problem of what does it mean to think about technology, you have to think about many different types of technologies that span human history and different usage, everything from a hammer to the stone that functioned as a hammer to street signs that function to tell us things, versus different social technologies, like the classical Foucault example of the panopticon, a technology used to instill fear, instill control over a large group of prisoners while reducing labor costs for guards. Or even something like the internet, something like code. And as I'll turn to after this little dive into the philosophy of technology, synthetic biology. So if you walk around on the street and you kind of just ask people, what is the point of technology? Usually you'll get something like technology is just a mean towards an end. It's just a tool. It's instrumental is a word you'll hear thrown around. Like a hammer, the classic example of someone just hammering a nail. All the hammer is just a means to get the nail into the wall, maybe to hang a picture, maybe to build a house. But there's also technology that's really interpretive. There's statistics, which is a technology that Karl Marx used in his famous work, Capital, to reveal a suspicion that he had about the role of the economic system. Or technology might be constitutional in the sense of the subway design in a particular New York City bus, in a New York City beach. They designed the overpass to be so short that city buses could not actually go to the beach, thus segregating the beach. This example was recently brought up by the transportation secretary, actually, as an example of a kind of constitutional moment of structural violence of technology. Technology is also emancipatory. It's freeing. We think of the government printing office being the first moment in the United States history where people were able to read and engage politically no matter where they were in the country. And, even today, we're still trying to understand what types of technology machine learning is and the ethics of AI, which I'm not even, going to talk about today. And so let me give you now a couple examples of some simple ones in the synthetic biology world, just to start thinking about dual usages. So one example is a company that's producing these very tiny chips to do DNA sequencing. So on the one hand, DNA sequencing is fundamentally important to the future of medicine. We know that cancer therapies are being designed to be personalized to tumors. And the better that we can sequence particular genes of patients on the fly, because it's cheap, because it's portable, because it can be shipped all over the world, will be fundamental to improving simple things like reducing suffering of chemotherapy treatments. But there's also another side of, if you sit back for a moment and think, what can a tiny chip who could sequence your DNA for like $5 do, right? I mean, you can imagine some really weird people who will bring these to universities and put them all over the place and sample people's DNA. You can think of different cases where this might be used for surveillance, where the simple cost reduction and wide availability creates new kinds of problems. Another example that I was mentioning was my own work, where we're creating a computational drug discovery platform, is what it's called. But on the other hand, you could call it a molecular optimization platform, where you can optimize molecules to do whatever you'd like them to do. Maybe instead of targeting the protease of the coronavirus, we might target fundamental proteins to the human biology. So instead of creating drugs that are good, we create drugs that are bad, or I guess in the Greek sense of the word, we create the poison. And this has recently been talked about, actually, interestingly enough, in Nature, and they have this great example of this. So this was a kind of speed whirlwind tour of ethics of biology or synthetic biology and the dual use problem. But now I want to give you three kind of proposals of why these might tip the scale in a different direction, and why AI might be bringing in a new kind of paradigm of thinking about risk, of thinking about how do we balance the things that we might not know about technology, about uses that might not be the intended purpose. So I have three proposals for you that I'm going to walk you through. So the first one is going to be a system for probing models. So I call this model probing for awareness. So one example is in the drug discovery case, one fear that we have is we don't want people to be able to use these platforms to develop toxic agents, to be able to develop, say, peptides that are dangerous. We don't want them to develop rapidly drugs in that case. But if we have the models, we actually can know what those models can do. So in a sense, by having the tooling available to you, you just need the funding and the care and concern to go through and think about, well, what are the toxic agents? What are the toxic effects? And when you create these models, there's techniques in differential privacy, for example, that you can use to, say, prevent models from going places where you don't want them. You can add noise to the data in certain ways to try to obfuscate different regions that you might not want. You can create surveillance programs that do high throughput screening for counter agents. So in the same amount of money that we do screening for COVID, we can also be doing screening against certain known peptides that models have produced that we know are toxic to try to create antidotes to them. And from my own work of doing high throughput screening, all you need is a couple supercomputers and a couple people invested in the idea that security just also means screening our tools in the same way we screen our ways that we produce our knowledge. So the second example, I think, is probably the most important one, and one I've written about more extensively, is this idea of cloud labs actually being a democratizing moment. So I didn't include any numbers here, but if anyone in the audience is studying development in the computer sciences and software engineering and AI in Africa, you'd actually know that the Google Collaboratory, an online platform for being able to access reasonably high performance computing devices to train ML models, is actually huge across the African region in the education of software engineers and future ML scientists there. And you might ask why? Well, because it's able to separate the infrastructure required, the expensive technical infrastructure required, that doesn't just require money, but requires the political and the social organizations to actually implement it, which currently is only available in kind of major developed countries. But it allows them to access this over a wire, over just a wire that gets connected. And all of a sudden, you're able to see a rise in software engineers because people can access computer resources on very, very relatively cheap hardware. And so I think that the same argument can be made for the idea of cloud labs. So Dr. Ramanathan was talking about the kinds of robotic and autonomous laboratories that we might be developing at Argonne that we're using at Argonne. But it's also reasonable to say that the future scientists in different developing regions are going to be able to be using these platforms in a way that doesn't require major structural change. And now I should bookend this. This is not to say I don't believe in any type of major structural change, but I do think that this is a quick solution to being more inclusive in the sciences. And so just to talk a little bit about the kind of platform that we're building out at Argonne, I mean, the idea is, you can imagine, is quite simple. So you have a bunch of different kind of research programs that are just kind of like programs you might have at a major computing facility, who all get time in this laboratory. They get time, they get reagents, they get tools, they get cells, they get samples. And they essentially are able to push these out to the lab. The lab does this kind of internally, runs. There might be a lot of debugging that needs to be done. And then the results kind of get pushed back to them. And just like in an API, and this might involve different exascale computing systems. But in a sense, what this does is this opens up the possibility to, one of the marketing videos is someone at a coffee shop coding up an experiment on E. coli. But just because it's a coffee shop doesn't mean that it's, has to be there. I mean, this really can be done anywhere. And it's then just a point of trying to be inclusive about it. So you might ask, well, what does inclusivity have to do with what I was talking about earlier, the dual use problem? Well, I'm happy to say more about it, but one way to think about it is there's certain unknown unknowns in the world. And there's certain social conditions, such as the vaccine hesitancy that Mike mentioned earlier. And one problem is getting the knowledge out to people wherever they are and meeting them where they are to share that knowledge. I could speak from experience as a first generation student. My own family wasn't very interested in getting vaccines, or by not interested, I mean probably passionately against it. But of course, because I am in their household, I am there to explain, push, urge, and meet people where they are. And the same way that in different countries, by being able to connect scientists to global infrastructure, you're able to extend the reach of the kind of epistemic knowledge that's available to us, where we are, and try to reach others. And so by reaching others and by sharing the knowledge, one thing you're doing is increasing the overall diversity of thinkers. And by increasing the diversity of thinkers, the number of unknown unknowns becomes more known, I suppose. And so that's a very important component. And in this work too, there's also then the security problem of, well, if we are producing peptides that might be dangerous, if we're producing genetically modified organisms that might be dangerous, do we want that happening all over different universities' labs where, there's different security protocols and things? Or do we want an automated central lab where, I don't know, maybe we put it underground or something where it's isolated and there's some secure protocols that, all experiments might get screened. So maybe there's some sort of closed loop paradigm. But, there can also be some type of, human in the loop idea going on. And the last kind of proposal that I want to throw out there is a bit more on this privacy preserving notion. And, there's this famous article from Wired from Chris Anderson in 2008 about the end of theory. And there's been a lot of debate around what's called the sort of epistemology of science about, is the way that we're doing science changing? When we make models, we're not really making theories in the classical sense. We're kind of making these objects that are in our computers that are stored in binary. They can make inferences just like our theories that they can do, but they're not quite as interpretive. I mean, they don't travel very well, that's for sure. If my colleagues make a model and they start asking me about it, I say, I have no idea. Why don't you go ask the model? So this led, one philosopher to call it alien reasoning, because it's like, well, I don't know. You need to go ask it instead. But this idea of asking it is also, very interesting. In the ethics of synthetic biology, you don't hear about this too much, but in nuclear studies, you do. I mean, why is it the case that the how nuclear reactions occurred, chain nuclear reactions occurred? Why did that ever escape, the field of the University of Chicago? Well, you start printing stuff out, and you print it out in language. Well, everyone can read that, at least everyone who might want to maliciously use it, and it's pretty easy to copy and send around. But there's a deep question, I think, about, is that the case of a model-based science? Is that how model-based science would travel? I mean, one could say, I guess, if we all share our training data, which maybe we should, then of course we can all build relatively similar models. But we can also be more thoughtful about it, and we can try to train models that might be sensitive. We can make models that are, that are classified. We can make models that are not classified. And there's a funny example, just kind of if you work close to the Department of Energy, you realize there's this little training snippet that always says, that I find interesting, that if you bundle together unclassified things in a certain way, that it may still become classified. And I think that there's something interesting to be said about this in terms of security risks and thinking about synthetic biology. So out of the three proposals, I want you to maybe draw a single thread from this. And one is that we're going to increase the possibilities of threat identification, that if we're having computational platforms that are screening and doing discovery, we could also have computational platforms that are doing risk mitigation, that are doing risk prevention, and that are helping us think about those risks. I think that AI can help us decouple hazardous materials using the cloud laboratory. I think that it can increase global inclusion and bring other scientists into the game, which might increase ultimately social adoption. And I think also that there's an intellectual property question in all of this and about the state of knowledge and how it flows. And I think that in other domains, AI is a major source of social awareness, which is where my current work is mostly, is in thinking about how counterfactual analysis is unlike any bureaucratic counterpart we've ever had. If you've ever accused someone of sexism, of racism, and you try to ask them, you try to prove it to another person, a bureaucratic institution can weasel its way out in a way that an algorithm can't. If you ask an algorithm a counterfactual question, it's going to be what it was, no matter what. And this is why I think AI is a powerful tool for social awareness. So now on to my final point, which is just about the question of ethics then, which is what is our role as scientists and how do we think about it? So I hope, I didn't explain it too much, but I hope you could see that synthetic biology has extreme dual uses. That AI probably can be used to mitigate certain ones, maybe push the scale in one way or another to prevent what some might call for a moratorium on synthetic biology, which is also a discussion. But the question remains still of, well, how do we decide and who do we decide? I've given you some evidence that we could push the scale, but does it push it enough? So I want to turn to a quick moment to think about democratic decision-making. And I want to draw on a distinction made by a Northwestern philosopher, actually, about there's two components to making a decision in a democracy. On the one hand, there's the answer. So, I might ask, what are the rules of nature and what are the rules of life? What is the relationship between inflation and policy? That one I'm still waiting on an answer for. How do masks impact infections? Insert any kind of policy question you might have. But there's also the other component, which is the social component, which is community, which is care, which is trust, which is being responsive to the people. And both of these together are required for anything to be done. So you might say during the COVID pandemic, we had the right answer to how do we vaccinate people? Well, we found it, we made a vaccine. But one thing we did not focus on was a social integrative component. We didn't focus on how might this vaccine actually impact people? How might people actually trust in it? How might we be responsive to the kinds of concerns that people have, or to the kinds of community building that might be needed for those vaccines to actually impact and do something in the world? Another example is, what if tomorrow someone says, I came up with a geoengineering solution to climate change. If we just release this particle all over the world, climate change will be solved. Well, that's great. But if they can't convince anyone to do it, well, then it doesn't actually solve anything. And so I think it's very important for us scientists to keep in mind there's two things that must be taken care of. There's always the right answer. And I put ethicists over here because I do think there's some benefit to moral reasoning, to thinking about what is just, what is right. But those are just answers to questions. Just because something's right in the capital R sense doesn't mean that it might be right for that society, for those people, for that time. What's right changes. And so I think it's very important that we take the questions that the public are asking, food security, water security, equality, climate stability, wildlife, and we listen to the kinds of questions they have. And we tell them what are the things that they can know? What is the spectrum of possibilities that we scientists can know? While at the same time, we also respect, listen to, and build the kinds of institutions that are the glue for people to trust in things, for us to be guided, to be regulated, to be just, to be practicing equality and global inclusivity. And so I don't want to say that it's ethics that's the answer. And I don't know if it matters what ethics has to say, but it matters at least insofar as we work together on building the public sphere and the civil society that will work for the future of synthetic biology. And so to summarize my kind of argument here that I'm making for you is, you know, synthetic biology poses some deep, wonderful solutions to some of the most pressing issues in the world. But it requires two things to work: the scientific answer, as well as the social integration in society for it to be an answer. And synthetic biology does have major concerns.
What happened?
Austin, are you frozen?
I think we froze him.
Oh, he's so close. Is he in his office, Arvind, or is he?
I can message him though. No, he's at home.
Oh, okay. Lately, it's happened occasionally because there's a lot of construction and other upgrades.
Yeah, he probably is.
Yeah, I think he'll rejoin, I'm sure. Yes.
Well, it does seem like he was getting to the end, and on his screen, he had some pretty interesting dilemmas.
Yeah.
Vinayak, I look forward to him coming back so we can grill him himself. But
yeah, I figured that sometimes a scientist and engineers talking about ethics is different than folks in humanities, social science talking about ethics, because our training, our experiences are different, right? So I think this would be very provocative discussion, I hope.
I think that discussion and that interaction has to happen, and I really.
There you are, Austin.
Oh, sorry, my internet is a bit.
That's all right. You can go back to your presentation. We'll wait for you.
All right, let me finish up the point. And so as I was saying, you know, I think that AI presents a lot of tools for us to invite others into thinking about the ethical dilemmas of it, and also of being mitigating factors. And so I invite the scientists in the room to think about ethics, not as something that's for them to decide, for the ethicist to decide, but as a collective experience. And we can do our role by providing the kinds of analysis of risk, by providing the data analysis of, you know, are these being used badly? How can we adjust what we do? And I think ethicists can increase their focus also on what it means to find answers and what it means to communicate those with the public. And so that's it. So thank you all for listening, and I hope there's some good disagreement, or I hope that means I did a good job, I think.
Very good. Thank you, Austin. Back to you, Sarah. I just wanted to make one comment that we wanted to have this event to have diversity of various kind. Mike being a senior member, then Arvind being at Argonne and U Chicago joint membership, and then Austin as the aspiring young colleague defining the future of the field. So, and we have Sarah bringing in anthropology perspective. So I'm just enjoying it. So back to you, Sarah.
Thanks, Austin. So I wanted to ask you a kind of high-flying question, which is if you could wave your magic wand today and solve one of the problems that you highlighted in your talk, what would your magic wand show us?
I think maybe you might know the answer to it, but the answer is I think the magic wand would be democracy. I think the magic wand would be
what?
Democracy. So
that's a big word. How does democracy
look?
So
let me tell you what I mean by this. What I mean is I think one of the biggest problems that we're facing is inviting people into discussions of knowing how to deliberate among people, knowing how to share concerns, knowing how to do kind of these basic interrelational important things for taking the kinds of work that, you know, scientists do and understanding how it can better serve people. I mean, I think that is kind of the ultimate problem in some sense.
So sorry, that's what you would like to see happen.
That's what I would like to see happen. I think if we had those institutions, those kind of deliberations, these kinds of town halls and things, I think the science would move a lot faster than even the scientists think. And I think also that people would actually be able to get their concerns answered and live in a world where they know the rules out, which is one of my favorite quote from a political philosopher, Thomas Cristiano.
Excellent. Well, we've had people with a lot of staying power during this talk, during these series of talks. So I think we should open it up to the masses for questions from the crowd, and you can type them in, or you can just unmute yourself and go ahead and ask them.
We're getting a few in the chat already. That's great.
Yeah, Chris Foreman is pretty excited about this kind of thinking. Chris, did you want to comment or question?
Oh, yeah. I mean, this is something I've been really interested in way back when I was a postdoc at Cambridge, and I set up these public forums, and I'd have like 100 or so people. Sorry, I'm in a place where there's background noise. And that was a lot of fun. Yeah, that was a lot of fun. And I didn't know what I was doing at the time, was reinventing this thing called deliberative democracy. I'd never heard of it. And I've learned about a lot of it since, and I've been quite involved in the US in setting up sort of national sort of approach to deliberative democracy. And for this community here, it's a really important way for scientists to engage with the public. And there are different kinds of town halls where you don't have a bunch of typically sort of white rich men at the front telling everybody else what to think. You break out into groups and you have really deep discussions and really powerful decision-making processes. And that can be a two-way street. It can really inform scientists in their research, and it helps the members of that deliberation really pick up that science and feel like they're engaged with it from the beginning. So I think those are two really important things: getting input from the public into the research direction, but also in terms of you know, helping the public pick up the new technology that's coming out at the far end of the research pipeline. So I'm really excited to hear somebody talk about deliberation when I didn't bring it up. So that's really great. Thank you for that.
You called it deliberative
democracy. Yeah.
So yeah, so that technique that Chris is referring to, for those in the audience who might want to Google it, they're called two things. They're called deliberative polls, and they're called mini publics. James Fishkin at Stanford University is the inventor of. He holds a patent on this, which is, I guess, an interesting comment. Is this idea of you gathering people together from different walks of life to inform them and have discussions with each other, and then ultimately vote on some sort of issue at the end. And I think, kind of going off of what Chris said, I think one of the most important things about these, for at least from my side, of like, as Mike was saying, we're at the beginning of an exponential growth curve on what synthetic biology is. I think that the most important aspect of these is communicating what's possible. Because by communicating what's possible, people can actually then say what they want. I mean, one major problem in the world is kind of alienation, where people don't even know the kinds of things that scientists can do, and then therefore can't even make the kinds of requests for the things that matter to them on the ground. Because, you know, the ability to have, say, a cell-free system that's producing you know, different medications or something is not even a thing that would be known, I think, to a lot of technology officials in developing nations. So I think it's important to share the kind of states of knowledge, not just for the facts, but also for the path that it takes.
I actually want to make one more comment on what you know, Austin and others were speaking. I think many of the funding agencies today are controlled by a few select folks. I mean, it's either through philanthropy, like Bill Gates, who basically donates much of his wealth towards these types of things. But at the same time, I think one of the things that is more important is to get inputs from the people that are on the ground doing work. So, you know, people in developing countries have very different challenges than what we face in this country or what we face in other countries for that matter. And, you know, having conversations, having deliberate conversations with them in terms of guiding how our scientific process might reflect the development of those, you know, goals that these people might have on the ground. None of the funding agencies actually take this into account. Everybody wants to transform climate, everybody wants to transform materials, transform this and that, and drug discovery and blah, 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 make it cheap, make it commercial, make it big. But none of it is actually taking into account what folks on the ground who are working you know, in these places actually have any inputs on. And so I think this is sort of important for the community to go after.
Eric, you're making a comment about the name synthetic biology. I know it's also referred to with other terms. Mike, do you want to say something about synonyms for synthetic biology?
Yeah, there are definitely a few different terms. Synthetic biology is one, kind of built on the idea of synthesis. Engineered biology is another. And actually, in watching Austin's presentation today, was just reflecting on the use of GMO, for example, on foods as a commentary, noting that it's no longer GMO, but the word is now bioengineered, if you look at what is now rolled out recently. And so I think that there are different terms being used. Some would say that they're different. And this has been a challenge for the field broadly.
And do you prefer the term bioengineered over synthetic biology? Do you?
I think I was intentionally specific with how I framed the beginning of my talk in the context of, as I imagine what synthetic biology is and how we might define that. So for example, I really look at it as the way in which we advance and apply our capacity to partner with biology, to partner with biological systems, to make what is needed where and when it is needed on a sustainable basis. And so for me, it's the idea of being able to facilitate synthesis in that space.
That was like a really academic answer to what we should call it. And so, I mean, for example, as an outsider, like chemical engineer could sound scary. I mean, it's like, ooh, chemicals, what are those? Like, I mean, pretty much everything is a chemical, but it sounds a little bit scary, but it's somehow entered the lexicon and it's not a thing in the way that GMO is a scary thing, even when there's lots of things like, plants naturally, I mean, anyway, that's the other thing. I think as an outsider, like bioengineering is just like another flavor of the kind of engineering we've done, and it doesn't sound so artificial, maybe. But
and I think it also came with the term engineered biology has been used commonly. There's the Engineering Biology Research Consortium of many synthetic biologists, which are connected and working closely with the government and policymakers to advance these capacities. I think it's tricky. I'd like to, Austin, I appreciate your comments, and I think the idea of deliberative discussions is important. I would also articulate that over its lifetime, there are definitely key ethicists that are a part of the conversation and have been since the beginning. Even the very oftentimes initial, synthetic biology activities are directed back to an engineering research center that was funded by the National Science Foundation called SINBER, and integrated very directly with that was a human practices element, which was front and center, led by Laurie Zoleth. And in fact, when you look at the International Genetically Engineered Machines competition today, human practices and ethics is, of course, baked in from the beginning, because I think we need to have these deliberative conversations and that they are occurring. I agree, not everyone knows all the science, but I would also say I think that this community is very engaged in kind of having those discussions and wishing for them, in the sense because we think the technology can lead to solutions to societal problems, even local ones, Arvind, as you were mentioning, that can address key problems of society in a just and equitable way. How to get there is a challenge, right? How do you enable the governance that's necessary? How do you enable the technology? How do you open access and education to all so that the field can be practiced? These are open questions that we don't have answers to yet.
Let me ask Arvind, if artificial intelligence is like has there been scary connotations of that, or how did the field get around that?
I don't think the AI community has really gotten around. There is quite a bit of fear mongering amongst the community, rightly so, because you can see a lot of things that have happened in the past. But there have also been very strong voices that have been raised. Tinmit Gibru and others who are actually like saying that the Google's algorithm that it uses for AI are biased. And the nice thing is you have ways to actually quantify this in AI, right? You have ways to capture what constitutes bias, what constitutes, wrong answers and so on. So there is little, there is less apprehension in terms of like trying to say how AI could potentially be, fear generating. But there is definitely concerns amongst the community in terms of, how this is going to affect our day-to-day lives and how it impacts certain things, especially when it is used in law enforcement, when it is used in, credit ratings and history and, maintaining those sort of decisions. I think there is a lot of fear in terms of how that is applied. Austin, you might have more points on how AI.
Yeah, so I first want to just go back to the comment from Eric and just respond briefly. I think on the one hand, we can point to names of things being fear and creating fear, but I think as scientists, we should have that analytic muscle to look past what is fear and what is public fear. I think that we should not reduce citizens to kind of these autonomous bots that might experience an emotion and not be able to think. On the one hand, it might not be they were not invited to think in the first place. And I think so it's an important thing to push past, I think, a little bit of the kind of over grouping the public into a simple lump of public, and instead like thinking about the ways in which they've not been included. For example, I know Mike Jewett first, at your team and everyone would be more than welcome for these types of deliberations. But I think too, it's not enough to just say there's some ethicists kind of around. Who are these ethicists?
No, I baked them into my laboratory. I've had ethicists sit in my laboratory and work with us. So Lori Zoleth has sat in my group and got to know and help us understand and ask good questions. So it's not just, oh, there's this one thing. It needs to be a deliberative and intentional conversation. For example, years ago, I participated in multiple meetings, including a symposium here, with the religion department. For example, I've spoken about synthetic biology at the American Academy of Religion meeting and having discourse with both them, but scholars, but also the community outside. But it takes all of us. And I think that's the point. It's not just one person here or there. I think that as we imagine what the benefits and the risks are, we need to continue to engage all.
Yeah, no, right. And that's exactly my point. I just wanted to make sure it was clear to the audience that, I don't think that what is like having ethicists around in a sense is a great thing, and it's better, of course, than not, because ethicists are trained in asking good questions. But to me, it goes deeper than just pointing to people to kind of say if something's right or wrong, but it's inviting all of those who are out there into the discussion, into the room, and because ultimately, otherwise, then we're just pointing to different groups of people. And so I'm happy to hear it. Of course, Mike is here at this event in the first place. So of course you're already 5 steps ahead of this deliberative process, right? So
I do think one of the major challenges is that the truth is that future is unknowable, right? So we are developing capacities to partner with biology. And what does that mean? When people developed processes to automate the construction of the automobile so that all people or new peoples could access that at lower costs, people did not imagine or could not have predicted how that would have reshaped cities from a cultural perspective. Similarly, the advancements in silicon chip building wouldn't have predicted the social dilemma that our children face by applications on cell phones. So certainly we need to have those conversations so that we can deliberate together, but we also, just in honesty, can't predict exactly what all things will look like. But I think that's why we need to engage very closely with others and figure out how to do things in a way that is just, how to do things in a way that is equitable, how to do science and synthetic biology in a way that we consider the risks that might arise from development of synthetic biology applications, right? How do we do that? And we have to engage that as a community.
Arvin, is that a hand raise or a stretch?
I would also say part of it relies on us being honest, right? Mm-hmm. There's such a huge element of being a scientist. So we need society to trust in us in some sense because we are being honest back. And so there's also this partnership that has to exist. And that, of course, arises and emerges from the opportunity to deliberate. So, and to just have the conversation, not, in a very like community-centric way, right? So
I was going to ask Sarah, you have a very unique perspective. You straddle on one side, SynBio folks truly deep into the science. You're in the anthropology department, you work with all sorts of folks, you visit different parts of the world. So how do you see the science and technology interface with public? And what sort of feedback do you get, if you'd like to share that as a part of this conversation?
Yeah, I mean, like thinking about the study in Kenya, where we brought the freeze-dried biosensors that Mike was talking about in his presentation. We didn't have to call it synthetic biology to say like, hey, these will turn yellow if there's fluoride, and what do you think? And then understanding what the implications were. Like, if it works, you can call it, whatever you want. Mm-hmm. What I find often is that there are very fancy fixes that get exported to places that the fixes don't work. And, if that's even, buying an ambulance for a village and then it not being able to work because, the tires are bad or some small thing. It's like a mismatch between technology and the setting. And that's bad for everybody in 5 to 12 different ways. Where I can see, why I have put my efforts in, minimal efforts, but where I've put my translation efforts into some of these bioengineered technologies is because they seem to work, and they're cheap, and they work. And so, I mean, it's like a famous last question, like what could possibly go wrong? I say that far too often these days, and ironically, but I mean, this can work if this can be a good fit because it seems to work. But I mean, there's more to find out and see if it actually does. Because there's so many fancy things that seems like can save the world. Water out of air, and energy out of fecal material or whatever. But does that actually happen?
And I was going to say, what's great about your example, though, there is very clear who's deciding, though, if it works, right? I mean, it sounds like, when you're giving these biosensors out in Kenya, it sounds like the location, right? I mean, there's a sense to which the people have decided that it works for them, right?
Right. And so what we call it in anthropology, we call it community-based participatory research. And I think that might be something like Chris Foreman is talking about with distributive democracy. It's where you say hey, what do you think about this? And how do you want to try to see if this works? And then we do it together, rather than outsider come and say this is the norms, or this is the standards, or this is our definition of success.
I wanted to ask a slightly different question, but we'll come back to this theme, Sarah, if you feel this is not an opportune time. I was going to ask Arvind, for example, one of the other areas that's fast emerging is quantum computing, right? So you have a different way of computation that's not based on your classical silicon. And from what we understand, that those kinds of quantum computing models are more apt, or more relevant to the complexity of biology. Do you see from your vantage point, is this somewhere in the next few years? This is still tomorrow's technology in the future?
A lot of people in the group are working on quantum computing technology inside the lab, right? I mean, personally, I think we have not we have not really explored a lot of questions that come from quantum biology, quantum sensing or quantum network technologies into our research per se. But we know of companies that are doing a lot of work with quantum computing sort of things for drug discovery and protein interface design and everything else under the rock using using these quantum computers. There is some more ways to go in terms of adopting these technologies directly into the biomedical community, but I'm sure it's going to take over some of the traditional routes of how we have been solving biological challenges. So much so, I think the Department of Energy has invested some of its money this year and next year in terms of looking at the promise of how quantum technologies can be really useful for quantum computing technology can be really useful for various applications, mostly in the battery world and places like that. But I think we'll see a lot more of this, technologies foray into our worldly applications, including biology, to a great extent, because I think there is promise in them. And in the last 10 years, how the technologies have really scaled up, and especially with the ability to program things using GPUs and the computing technology advances, there is real promise in that area in terms of achieving some real good breakthroughs. And if those things happen in the few years, and I think they're imminent, then I think we do have the ability to apply them for more challenging questions in biology, because many of the problems that we try to find algorithmic solutions in biology are known to be NP hard in a complexity theory sense. These are problems where we cannot solve them in polynomial time. So the number of combinations are quite large, and so it's very hard to solve them using traditional computing. But that's not a problem for quantum computers. They actually work with probability and statistics, so they're pretty natural when it comes to solving problems there. So you will see a lot more of these problems becoming more and more solvable. And if that happens, I think we'll be reaching a point where there'll be even more exciting stuff that can come about with automation, with these technologies playing a role in how they guide the design of experiments even. So
Vinayak, I wanted to ask you, as someone who's seen a number of trends isn't the right word, a number of movements, a number of developments in science. I mean, artificial intelligence is certainly one of them, so is synthetic biology. I mean, what are some similarities and differences either between the two of them, or if you would compare them to other big science movements?
And I suppose, I may not be the best person to answer, but for me, I think complexity comes to my mind. I think both are complex systems, both are multifaceted, multivariable phenomena. And I think it's not very easy something to predict where things would go. But I see an opportunity for both of them, for synthetic biology to influence AI, ML, and vice versa. And I think that's where we are in right now. We are in the convergence. So the part of the theme of our talk was convergence. Are there some common denominator that we see between the three fields, data science, synthetic biology, and ethics? And I made a comment in the comment section, which is that, over the years when I followed nanotechnology first, before that microelectronics, people often say that the science and technology might change, but the ethics remains the same. And so that was my question too, are there some common ethical consideration which transcend different science and technology? And this is probably a question for you, Sarah and Austin, than for me.
Well, I am not an ethicist, but I will say that the ethics don't stay the same, because with new technologies, I mean, new possibilities, new ways for things to go sideways comes up. So while we might have, universal, long-lasting principles of non-beneficence and non-harm and goodwill towards humankind, the manifestations of that can be very, very different.
Yeah, I think I would come in and say one example of that would be the human rights movement has actually changed a lot of the considerations around from what used to be a kind of majority. I mean, in some sense, you know, in some sense, one used to think of ethics as coming directly more from ethicists, which was generally more of thinking about minimizing harm and things. But nowadays, there's a relatively strong, at least in a legal basis, of rights-based approaches, which protect against different things, protect against different issues, and do so in different ways. And so maybe so I guess it's kind of a roundabout answer. The ethics changes, I think, in the way Sarah is pointing to, but there's also then the kind of institutional practices that push that in a different way.
Yeah. I'm going to take a page from literature. So right now I'm reading this book by Ian McEwan. It's called Solar. I think it would be of interest to many people on this call. It's about a fallen, kind of unlikable Nobel laureate. He is pitiable and hilarious in many ways. But he has a perspective on these epochs in science. And he stands before a crowd where he is announcing how solar is going to revolutionize, and he draws on fossil fuels, like the revolution in fossil fuels 100 years ago. And I would say to you, fossil fuels would be this thing. Or if I would say, you know, 50 years ago, and plastics would be this thing. I mean, it feels to me that the topics here today are going to be these things. They are, although they're front and center in the people's on this call's mind, you know, my mom and dad don't know about these things really. But I think that in 20 years, my mom and dad will understand bioengineered fuel cells or what artificial intelligence means. And I guess it's just all this is to say is that we are on the cusp of really transforming, and transformation can be both painful and scary, but it can also open up so many new pathways that it just makes me really proud to be a part of the Northwestern community and part of the larger scientific community where we're able to have these dialogues. I think that might be a good place to leave it, unless there are other concluding comments from each of the other panelists.
Yeah, so how about some wrap-up comment around the table, Sarah? Starting with Mike and then Arvind, Austin, and then you.
Yeah, no, I would just kind of amplify some of Sarah's comments. I think it's really terrific to be able to be here today with this group of people in this place and to kind of begin additional discourse at a broader Chicagoland area that I hope really grows. I think that products from synthetic biology will continue to enter the market over the upcoming years and decades. And similarly to the ways that many of them are in products, many biotechnology products could be in things that we use every day that we don't know about. For example, you know, subtle lysine in laundry detergents is not something that a lot of people think about all the time, yet it's had a transformative effect on, you know, being able to get that tomato sauce stain out of your clothes while still washing your clothes on cold water and saving energy for the planet at very large scales. I think that products made by biology and bioengineered systems and synthetic biology systems will continue to expand. My wish is that we as a community can have conversations to ensure that that's done in a way that is just, that is done in a way that is equitable, and that is done in a way that helps people solve local problems locally. And I think those are some of the themes to me that I heard today, all enabled certainly by technological innovations and broader activities, and hopefully we can bring some of that to bear. So thanks again, Vinayak, for the terrific opportunity to be here and for organizing the symposium, and to Sarah for moderating, and just very grateful for all of that.
Thank you very much. Arvind, your thoughts?
So yeah, thank you very much for this opportunity to speak here, and a lot of fun to like interact and hear perspectives from different people and try to understand what's going on. I think apart from the fact that we have what Mike was saying in terms of things that are like slowly coming into the market and what's happening with synthetic biology, I think automation can also help a lot of things. Not only we as scientists, but also as tools that can help education, tools that can help how it can reach people that are not used to seeing these sort of things in the real world, ability to do experiments that potentially wouldn't have been possible before. Instead of having a lot of training in terms of like learning how to do experiments, this is about bridging that demographic gap that exists in terms of how to do experiments automatically, how to design experiments automatically and meaningfully. I think that brings into the foray of like newer techniques of teaching and methodology that comes into play, reaching out to potentially a large audience that's not even trained in biology in the first place. Like when I came to the country, I didn't learn any biology in the first place. I mean, I had no biological background, and that was mostly, in my grad school, I had opportunity to learn from like really good teachers, and and hopefully people are curious and they can actually start using these technologies in ways that they can improve their lives, potentially leading to like more innovative solutions in terms of drug discovery, reducing costs, making it more automated, and making it more reproducible. So thanks.
Thanks, Arvind. Austin?
Yeah, so again, thank you all for inviting us. It was wonderful to act not in my role as a scientist, but to talk a little bit about my other research area. I think one takeaway for me is reflecting on a little bit about Vinayak's question still ruminating in my head, and of Sarah's comments about how change in ethics occurs. And, I do think that it's time to reopen a lot of those ethical questions in synthetic biology, given the new technologies that we have available to AI. I think that there's a lot of new technology out there that does bear on others, right? I mean, the traffic light changed a lot of the ethics of how cars might go around. And so I think it's a good time to do a lot of good intersectional work and to think about, maybe how things might be differently if we make leaps and we make changes and we do things inclusively and we use computers also to address the worries that the publics might have. And also too, to recognize that there's amazing research going on by amazing scientists who are actually trying to do good work and maybe focusing on, let's get it out, let's share it, let's hear people's thoughts on it.
Sarah.
I appreciate all of you. I'm gonna just leave it to that.
Any final comment from you, Sarah, on the technical side of what you do?
On the technical side? No, I think we should look at this recording in 10 years from now and giggle about all the things we knew and all the things we didn't know, because the world's changing.
All right, so before I make a few comments, I want to introduce Kamran, who's our Shine Director of Operation. He is instrumental in organizing this event. You want to say a few words before I close the event?
Yes, first of all, thank you very much for attending this seminar. It was like really, really fantastic. Thank you. I just want to add a final comment. 25 years ago, I was a graduate student at Northwestern University. I got my PhD at Northwestern from the group of Professor Razegi, and I was totally a science guy. And then since then, I spent about 20 years in Silicon Valley, moving more and more from science and technology into what my colleagues used to call the dark side of marketing and business. One of the very interesting things that I found out in this journey, and Austin, you're going to see this if you ever get a job in Silicon Valley and go there, is how much the future of science and technology is driven primarily by finance rather than the science itself. I'm not saying this is a good thing or a bad thing, but that is how the world evolves in Silicon Valley. Everything is viewed from a money point of view, from a finance point of view, from a business point of view, which is quite different from what we are used to. And I can tell that because I used to be a scientist 25 years ago, and I evolved into a businessman throughout the years. And it's like looking at something from two entirely different perspectives and seeing two entirely different universes. And Austin, especially you, as you go into companies and corporations, and as you like go through the next 25 years of your life, just be aware that what we look at things from our scientific perspective is an entirely different thing from a business point of view. And I hope that in future seminars, we can invite like captains of the industry, like CEOs and presidents of companies in Silicon Valley, and they come and tell us how they see things from their point of view. So just an observation of leaving Northwestern and working in Silicon Valley for 20 years, this is like how it is. Excellent seminar. Was fantastic. Thank you very much. So.
Thank you, Kamran. I don't want to repeat the thanks, but I must, because this is an event I was looking forward to for many weeks. And yeah. I realized that each one of you have so many opportunities to give talks and be elsewhere, but you came here for this forum. It really means a lot to all of us, because it's a very unusual program that we do. We actually relish success of other people, and that's not very often that happens. So here at Nuance or Shine, we celebrate when people publish their publication or file patents or do innovation. I just want to add one quick note about the cross-training or cross-fertilization. We actually have a nano journalism program where we take journalism or communication students and we house them just the way Mike mentioned about some of the other outreach. And we want our people like in journalism and communicators to learn how creativity of science work. The topics are so complex, right? Synthetic biology, CRISPR, nano. And so we want our common citizen, our neighbors to learn about science, innovation, technology. And this is just a small step that we're taking. The other connection I think we have in the region is, of course, we have Argonne National Lab, other educational institutions, but we also have museums. And we do a lot of outreach with the Field Museum, Science and Industry Museum, as well as Chicago Botanical Garden, which has a fantastic collection of leaves and other fauna. And so this is an opportunity for us to really cross-fertilize. When I first came to this part of the world, I was told that there are only something like 2/3 of the days are cloudy in Chicago, and there's a very little sun. Why are you going to Chicago? And usually 30 years ago, people used to fly from West Coast to East Coast, and they didn't stop in between. But now there's a tremendous activity in the Midwest. A lot of people are specifically making a trip in the Midwest. And then that's a testament to a lot of you who are innovating in not just science, technology, but all the other fields. And we really want to thank everybody to participate today, because this was an unusual event. We're taking an emerging technology of science, as in synthetic biology, combining with AI with the ethical twist to it. So I like to think that the conversation has just begun, and I hope to see more of you in many different capacities on campus, in Chicago region, on Zoom meeting and elsewhere. So I'm going to hang around for a few more minutes, but I want to again thanks everybody. And Sarah, if you have any other comments. So we're good. Thanks everybody. Relish the spring that's in the air. Thank you, Katie, our whole team from Nuance Shine, really for pulling this together. Jordan, then a lot of names involved. So thanks everybody. Bless you and enjoy the spring and hope to see you soon.